We'll answer as many questions as time allows. It's now my pleasure to introduce for opening comments and introduce our first two presenters, Dr. Ethan Stier, Associate Director, Lifecycle Management and Immediate Office, Office of Clinical Pharmacology, Office of Translational Sciences, and CDER. Please join me in welcoming Dr. Stier.
Good afternoon. Welcome to the SBI webinar on the finalized guidance bioavailability studies submitted in NDAs and INDs general considerations. My name is Dr. Ethan Stier, and I'm the Associate Director for Lifecycle Management and the Office of Clinical Pharmacology, FDA, and I'll be providing the opening remarks to today's discussion. First, I'd like to discuss what's included in the guidance and what's not included in the guidance. So, of course, the main focus of our discussion will be on bioavailability studies conducted in INDs and NDAs. However, for bioequivalent studies used to evaluate generic drug applications, or ANDAs, will not be covered today. Please refer to the guidance, and also there's an SBI webinar about this guidance on the web as well. Although the Office of Clinical Pharmacology reviews biosimilar data, that guidance will also not be included in today's presentation. Please refer to the guidance attached. And now I'd like to go over some brief regulatory history highlights. So this guidance was first published in 2003. About 10 years after that, it was revised and published for public comment. Also at that time, generics were split off. At that point, they were actually combined with new drugs, so they were split off into their own separate guidance, which I mentioned a slide prior. In addition to that, there were a number of important clinical pharmacology topics that were included in the guidance, including how to handle endogenous substances, a new approach for highly variable drugs that could reduce the number of subjects required for a comparison, an expanded modified release section, also a section about how to handle narrow therapeutic index drugs, as well as recommendations about how to carry out or how to evaluate alcohol dose dumping risk in formulations. And then in 2019, another draft version was published. And then this year, in 2022, the public comments were addressed and the guidance was finalized. I'd now like to quickly run through the agenda of the titles of the remaining presentations along with the speakers. So Dr. Dakshina Chilukuri will be presenting an overview of the bioavailability guidance. Then Dr. Jayabharathi Vaidyanathan will be presenting special topics. Dr. Kofi Kumi will be discussing pharmacodynamic and non-traditional endpoints. And finally, Dr. Akpo Edidiri will be discussing in vitro studies. Just like you, I'm really looking forward to the following presentations. Thank you.
Good afternoon to you all, and welcome to the FDA SBIA Bioavailability webinar. My name is Dakshina Chilukuri, and I will kick off the guidance presentations today with an overview of the guidance and give you a flavor of the general considerations in the conduct of the BA studies in NDAs and INDs. Over the next 30 minutes, I will cover the general aspects of bioavailability studies. I will cover topics related to the importance of BA studies and when these studies are conducted in routine drug development, aspects related to study design and the appropriate PK measurements. Additionally, I will touch upon the alternative approaches to assess bioavailability. And finally, I will cover the recommendations and thinking behind the recommendations for the fed versus fasted studies. So what is this guidance about? The guidance describes in detail the typical bioavailability studies conducted in support of NDAs and INDs. Among other things, the trial conduct details are given for both immediate release and modified release drug products. As we all know, the most common approach to determine bioavailability is the PK study, and specific details on the conduct of this study are described in the guidance. Other approaches in lieu of the PK study are also described, and some of these aspects are described by other speakers during this webinar. It is to be noted that this guidance does not cover biosimilar products and generic products. For generic products, a separate guidance is being issued by the Office of Generic Drugs. So why are BA studies important? On this slide are described the general requirements for new drugs from a BA perspective. For NDAs, studies are typically conducted to demonstrate the bioavailability of the drug product, or information is provided to waive such studies. An example of a relative BA study that we normally see during new drug development is a study that is conducted between the clinical trial formulation and the to-be marketed formulation. This example is covered when specific case studies are discussed during the Q&A session. For ANDAs, the requirement is to show bioequivalence between the proposed drug product versus the reference listed drug, or provide information that allows the FDA to waive such study. And for supplemental applications such as those where there is a change in the manufacturing site, etc., studies are needed to be conducted to bridge the post-change products versus the pre-change products. And some of those scenarios are described on this slide and extensively discussed in the guidance. So BA studies provide useful information related to the relative fraction of dose absorbed into systemic circulation. They can also provide information on the linearity of the PK and help evaluate effects of food on drug absorption. These studies also give insights into the properties of the drug substances prior to entry into the systemic circulation. So typically BA studies are conducted to either determine the absolute bioavailability or relative bioavailability. In the case of absolute bioavailability, the comparison is for an oral formulation versus an IV formulation, and for relative bioavailability, the comparison is between an oral formulation versus a different oral formulation. And as indicated earlier, there are two scenarios where formulation comparisons occurs during drug development. One is pre-approval and one is post-approval. Pre-approval includes the to-be marketed versus clinical trial formulation that I mentioned earlier. And post-approval also includes the post-approval changes to the manufacturing site or any other changes that trigger the need to conduct a bioavailability study. In this slide, in the next 3 slides, I will cover the general features of a bioavailability study. Typical BA studies are conducted as a crossover or parallel design, depending on the PK characteristics of the drug substance. For example, a parallel design study is used for a long half-life drug, where a crossover design is impractical. And the guidance recommends pilot studies for a variety of reasons that are described on this slide. I will not go into I will not reiterate these points here, but the purpose of a pilot study is to generally get experience ahead of conducting the pivotal bioavailability study, particularly things like to optimize the sample collection time intervals and to determine the length of the washout period. So these are some of the reasons why pilot studies are recommended in the guidance. So typically healthy adult subjects are enrolled in these studies. The recommendation, the guidance is that both male and female subjects should be enrolled unless there is a specific reason not to do so. The guidance continues to advocate the need to conduct BA studies under single dose administration under fasted conditions, given that these are more sensitive than multiple dose studies. But there are scenarios where multiple dose studies are warranted. The guidance recommends that the parent or the active moiety rather than the metabolites be measured in the case of bioavailability for new drugs under NDAs and INDs, unless it is not possible to measure the parent for analytical reasons. Some features of the study conduct are presented in the next couple of slides. The study design should be accommodated with an adequate number of samples per subject and an adequate washout period to ensure that there are no carryover effects. when appropriate. This guidance recommends that if pre-dose concentrations of more than 5% of the Cmax, then the subjects should be dropped from the PK evaluations. There are specific recommendations for data exclusion in subjects who undergo emesis in these studies. For IR products, for example, if vomiting occurs at or before 2 times this Tmax, and for MR products, if emesis is reported any time during the dosing interval, that data is to be excluded. Under measures of systemic exposure, both the peak and total exposure are measured, Cmax and AUCs. For specific therapeutic areas, partial AUCs can be used. For example, ADHD and pain, where the onset of the analgesic effect, for example, in the pain indication, the use of partial areas could be done, and this is covered again in a subsequent presentation during this session. The B criteria of the 90% CI for both AUC and Cmax is applied for when the BA studies are described in the guidance. So for the purposes of determination of bioavailability, relative bioavailability, the B criteria of 90% confidence interval for both AUC and Cmax to fall within the 80 to 125% is described in the guidance. As indicated earlier, BA is determined most commonly by a PK study, but there are other approaches to support bioavailability, and these include IVIVC. In the next couple of slides, I will describe in brief about these approaches, and again the subsequent presentation in this webinar will touch upon this in a little bit more detail. So the first approach, which is the alternate approach to support or determine BA, is this IVIVC, where a relationship is established between the in vitro attribute and something like a plasma drug concentration. So a relationship is established between the in vitro dissolution or in vitro release from a formulation versus the plasma drug concentration, either Cmax or AUC. And so this relationship helps to support the bioavailability and obviate the need to conduct a bioavailability study. In some cases, pharmacodynamic studies could replace PK studies, but these are reserved for scenarios where the PK determination is not possible, and where there is a well-justified PD endpoint that is available. So these are the two scenarios when this is acceptable. And other approaches to support bioavailability, comparative clinical studies. So there are scenarios where the PK approach does not work, and in here comparative clinical studies are conducted with definitive clinical endpoints to demonstrate the comparability of the formulations. A typical example is of a therapeutic area is the traveler's diarrhea, where the site of action is the gastrointestinal tract, and the drug products are designed to often have very poor absorption. And therefore, in such scenarios, in the absence of adequate PK concentrations, the clinical studies are conducted to demonstrate comparative bioavailability. Other in vitro approaches, such as in vitro dissolution, they are not covered in detail in this guidance, but we will have a short presentation about it later on during the seminar. F2 comparisons could be used for these in vitro dissolution comparisons. Next 3 slides, I will go over some key aspects of the considerations for MR products, modified release products. The guidance describes modified release products as either extended release or delayed release products. So it uses the terminology interchangeably. So if I say MR and ER in this presentation, I'm essentially meaning the same. So whenever the formulation is modified to either delay or extend and control the release rate, so the guidance describes all of them under the umbrella of modified release. So this guidance, when it was revised over a period of few years, the guide provided specific recommendations for the MR products for these three scenarios, which I will go over the next 3 slides. So the first scenario is the one for the development of an MR product given an approved IR product. For example, when 100 milligrams ER once a day formulation is being developed for a existing formulation of a 50 milligrams IR to be given twice a day. So for this sort of an example, the recommendations included here are the following. For a drug with linear PK, the recommendation is to compare the highest strength extended release product versus the IR product over the entire ER dosing interval. For example, here the comparison would take place between the 100 milligrams ER QD versus 50 milligrams IR given twice a day. And for drugs with nonlinear PK, single dose highest and lowest strengths ER versus respective IR strengths over ER dosing interval is expected. Because of the nonlinearity, the coverage of strengths is needed. And a dosage strength proportionality study, which is described in the guidance, is required for this scenario. And single dose food effect study on the highest ER strength is also included as one of the studies that is needed for approval. And a steady state study to demonstrate the PK following multiple dose administration of the highest strength ER versus IR. So these are the 5 studies that are recommended, or 4 studies actually. It depends on whether the drug is linear or whether the drug exhibits linear PK or nonlinear PK. So these are the general recommendations as included in the guidance. The second scenario, which I referred to earlier, is this new ER product. When a new ER product is being developed and compared to an approved ER product with a different dosing interval. Again, the example is laid out in the guidance, so here specifically the ER new and ER old have unequal dosing intervals. So the recommendations for this scenario are same as outlined in the previous scenario. In this case, the reference product could be either the approved ER old or the existing ER product with a different dosing interval, or an IR product. So with this, the study requirements themselves are similar to the previous scenario. When a new ER product is being developed with and compared to an approved ER product with the same dosing interval, so these are the following studies that are recommended in the guidance. A single dose fasting study on the highest strength of the ER new product compared to the ER old product. And if the ER new and ER old, if the new ER product is being developed which has different strengths, then comparison of the equivalent doses using the highest strengths is recommended. And as in the other 2 scenarios, food effect study should be conducted on the highest ER new strength, the new formulation. And when the ER new strengths are not proportionally similar in composition, the recommendation is to conduct a single dose fasting dosage strength equivalence assessment study or a dosage strength proportionality study for the ER new product. I have not included a description of these 2 studies, but they're pretty well laid out in the guidance, and I refer to the guidance for the specific example for these 2 particular scenarios of dosage strength equivalence assessment and dosage strength proportionality study. I'll switch gears to the last topic that I'm going to cover here. This last topic is related to the fed versus fasted studies. So the guidance gives detailed recommendations in Appendix B for the conduct of fed versus fasted studies. Earlier I mentioned that the most commonly recommended study is the single-dose fasted study, because that is the most clean way of comparing two different formulations. But the Appendix B under the guidance presents 4 scenarios where specific recommendations are provided for when the drug is given, is recommended to be given under fasted and fed conditions. So I will briefly describe these 4 in the next 3 to 4 slides. The first scenario here on this slide is for NDAs, where the bioavailability is determined using a solution, IV formulation, or a previously developed formulation as a reference. And the recommendation here is that the BA should be conducted under fasted conditions, except when tolerability issues are expected. So again, this is similar to the scenario I mentioned earlier in this presentation. So the recommendation here is for a fasted study, unless tolerability issues are anticipated. And in addition to that, an effect of food on the BA, of food effect study on the new drug product should be conducted using a high fat and high calorie meal. This is to evaluate if this formulation, if food impacts this new formulation, and therefore the labeling recommendations can be developed based on that. And if the objective is to evaluate other meal types, then these other meal compositions, such as a low-fat meal or a moderate-fat meal, can also be assessed in addition to the high-fat and high-calorie meal. The second scenario, as described in the Appendix B, refers to the scenario when the reference drug product is labeled to be taken without regard to meals. Here, the recommendation is that the test and reference drug product should be compared under fasted conditions. In addition, the effect of high-fat meal on the new drug product should be evaluated. And alternatively, the bioavailability of the new drug product under fed conditions could be established by comparing the test product and the reference drug product, both administered with a high-fat meal. So the third scenario here is that if the reference drug product is labeled to be taken with food, then the test product should be compared under fed conditions. So the recommendation here is that if the reference product is to be taken with food, then the comparison of the test versus the reference should be done under fed conditions. And these fed conditions should be the same as the ones that are described in the labeling for the reference product. In addition, the evaluation of the effect of the high-fat meal on the new drug product, that is to test the test fed versus test fasted, can be useful if additional labeling is desired for the test product. Then this study could inform that that section of the label. And a recommendation that the guidance makes is that a 3-way crossover study could be considered, because it allows for the direct comparisons or relevant direct comparisons between the test fed versus reference fed, and also the food effect assessment, which is the test fed versus test fasted. So the 3-way crossover study could be considered for this scenario. And finally, if the reference drug product is labeled to be taken with food to avoid the tolerability issues in the fasted state, then the BA of the test product should be evaluated under fed conditions according to the label instructions for the reference product. So here again, because the reference product is being given with food to avoid tolerability effects, the recommendation is that the test product should be evaluated under fed conditions. So this brings me to the end of my presentation for today. Thank you for your attention. If there are questions, those can be discussed during the Q&A session after all the presentations are completed. Thank you.
Hello, my name is Jayabharathi Vaidyanathan, and I'm very happy to be here today to present the special topics in the bioavailability guidance. I'll discuss some of the special topics mentioned in the guidance. I will go through the approaches to determine bioavailability using case study and examples. This slide shows all the special topics in the guidance. I'll cover some of these, for example, combination and co-administered drug products, endogenous substances, drugs with long half-lives, and narrow therapeutic index drugs. Some of the other topics will be covered in the presentations that will follow. I'll start with combination and co-administered drug products. The guidance refers to a fixed combination product as two or more active ingredients that can be formulated as a single drug product. Generally, the purpose of an in vivo bioavailability study involving a fixed combination product is to compare the rate and extent of absorption of each active drug ingredient in the combination drug product to the rate and extent of absorption of each active drug ingredient administered concurrently as separate single ingredient preparations. A two-arm, single-dose crossover fasted study is recommended, and this study should use the highest strength of the fixed combination with matching doses of the individual drug products. This brings us to case study 1. No Pain is a combination product of two approved drugs, drug X and drug Y. Drug Y is recommended to be given with food for tolerability reasons, while drug X can be given with or without food. If a relative bioavailability study is needed, should the sponsor conduct the study under fasting or fed condition? If you were considering a study design under fed state, that would be appropriate, because if the reference drug product is labeled to be taken with food to avoid tolerability issues in the fasted state, then the bioavailability of the test product should be evaluated under fed conditions according to the labeling instructions for the reference product. The guidance recommends that the bioavailability studies for fixed combination product should include the measurement of systemic concentrations of each active drug ingredient. A confidence interval approach for bioavailability assessment should be applied to each measured entity of the fixed combination and its reference product. In addition, fixed combination drug products can exhibit different effects of food compared to when each active drug ingredient is administered alone. Therefore, the sponsor should assess the effect of food on the various active ingredients of the fixed combination drug product after administration of the combination drug product. And such evaluation of the effect of a high-fat meal on the new drug product can be useful to support labeling of the fixed combination. And generally, a single-dose high-fat food effect study design is recommended, and you can refer to the food effect guidance for more details. Now I will present an example of an immediate release fixed-dose combination tablets. This product was a combination of two drugs and was submitted via the 505(b)(2) pathway. 4 strengths of the fixed-dose combination product were being developed, and the strengths of these match to the approved reference product for each of the active component of the FDC. 2 relative bioavailability studies were conducted that provided bridging of the fixed-dose combination oral tablets to the reference product's findings of safety and effectiveness. Both studies were similar design, an open-label randomized 2 treatment, 2 period, 2 sequence crossover single-dose study. One study used the highest strength, and the second study evaluated the lowest strength and compared to the single doses of co-administration of two reference products given concurrently. The study measured the systemic concentrations of each active ingredient, and confidence interval approach for bioavailability assessment was applied for each measured entity. And the study results demonstrated comparable bioavailability of the two active components of the FDC to the individual reference products. Now I want to present to you another case study. This is for a fixed-dose combination tablet of two approved products, drug A and drug B. There is a potential for drug interaction between drug A and drug B. So if a relative bioavailability study is needed, which study design would be appropriate? Study design 1, which is a two-arm design of the fixed-dose combination versus the two drugs given concurrently. Whereas study design 2, a three-arm study evaluating the fixed combination versus the two drugs administered separately. The best study design in this scenario will be study design 1. We can also choose study design 1 in the case where drug interaction is unknown or not clear. Certain alternative study design could also be considered depending on specific situation. For instance, when there are no drug interactions between the components of a fixed combination consisting of two components, then a three-arm study design comparing the combination drug product versus the single ingredient drug products administered separately, as shown in study design 2, could be appropriate. I want to now move to a slightly different combination product where in specific cases, drug products are given in combination but are not co-formulated. And the main objective is to increase the exposure of one of the drugs, that is, the subject drug. The second drug, the booster drug, is not intended to have a direct therapeutic effect and is given only to increase the systemic exposure of the subject drug. Now, when both the subject drug and the booster drug are new molecular entities, the bioavailability of each should be determined individually and when administered in combination. Now, and when the combination, which is again not co-formulated, includes a new molecular entity and an approved booster drug product, then in this case, only the bioavailability of the new molecular entity should be assessed, because it is assumed that the bioavailability of the booster product has been previously evaluated. If for some reason bioavailability studies are needed for the booster drug, they should be conducted only with the booster drug. The subject drug should not be dosed with the booster drug in this situation. At this time, I would like to present another case study. Drugs S and Y are two new molecular entities, where drug Y is given to boost the exposure of drug S. The drug S formulation is changed post-approval, and there is a need for a bioavailability study. Should the bioavailability study for drug S include the booster? The answer in this situation is yes, if during development or after approval there is a change in the subject drug product formulation that results in the need for a bioavailability study, the subject drug should be administered with the booster for both post-change and pre-change products. The corresponding PK measures, including confidence intervals, should be determined and reported for the subject drug. In this scenario, generally it is not needed to measure the concentrations of the booster drug. Okay, so with that, I would like to now move to the second special topic of endogenous compounds. There are several drug products that can contain compounds that are also endogenous to humans. For example, various hormones and also compounds that can be obtained from the diet, such as omega-3 fatty acids and vitamins, etc. When the endogenous substances are identical to the drug that is being administered, it can be difficult to determine the amount of drug released from the dosage form and that is absorbed. And bioavailability determination can present certain challenges. First, for these endogenous compounds, there exist baseline levels of the target analyte without administering the test compound. And secondly, the baseline of the endogenous substances are not constant during the study period because they can be various homeostatic processes that can affect their production and therefore impact their systemic concentrations. Factors such as circadian rhythm, dietary intake influence the baseline of endogenous substances. To reduce the impact of this variability in the concentrations of endogenous substances, an alternative approach could be to enroll individuals in the bioavailability studies with low or no production of endogenous substance instead of healthy subjects. Or the inhibition of the production or downregulation of the hormone can be done by treatment. When the concentrations of endogenous substances are influenced by diet, restricting the dietary intake of the substance before and during the study could also be appropriate. In such situations, to achieve a stable baseline measurement, subjects should be housed at the study site for a sufficient period of time before the study and served standardized meals with similar content to that of the meal that will be served on the day that PK sampling will take place. So let's talk about a case study. NoGen is an endogenous product and subjects have pre-dose level of the drug. Are there any unique study design considerations for such a product? As we just discussed, the baseline levels are challenging for endogenous substance. Therefore, the main consideration for a BA study in this case would be adjustment for the baseline levels. So let's see approach for bioavailability determination for endogenous compounds. The baseline concentrations of endogenous substance should be measured in the period before administration of the study drug. Depending on the proposed indication, it could be advisable to subtract the time average baseline or time-matched baseline from the post-dose concentration of each subject. When using baseline-corrected PK analyses, the accurate measure of pre-dose concentration is important, and there should be multiple measures at pre-dose, and the mean value of the pre-dose measurements can be used as baseline for correction by subtracting this from the total concentrations measured from each subject after the drug has been administered. And generally, the pre-dose measures are average, and this average is subtracted from concentrations at each time point. But for the time-matched correction, baseline levels are obtained at multiple time points throughout, for example, the duration of the study, for example, 24 hours pre-dose, and are subtracted from the corresponding time concentrations that are determined post-dose. The baseline concentrations should be determined for each dosing period, and the baseline corrections should be period specific. And finally, the PK and statistical analyses should be performed on both the uncorrected and the corrected data. Let's see an example of a drug product containing two endogenous compounds. This product was a fixed-dose combination product consisting of a formulation containing two hormones. The sponsor conducted a bioavailability study using two individual reference drugs and the proposed fixed combination as the test product in order to bridge certain findings from the individual reference product. The study was conducted in healthy postmenopausal female subjects because it was expected to have less interference with baseline levels as compared to healthy subjects. Single-dose, three-way crossover reference replicated study design was conducted under fed conditions because of large intrasubject variability in PK of one of the endogenous compounds. Samples were collected at 3 pre-dose time points shown in the slide for baseline corrections. And this average was subtracted from all the post-dose concentrations to correct for baseline. And the PK parameters for both baseline adjusted and unadjusted levels of all the active components was determined. I will now move to the next special topic, drugs with long half-lives. And I will present a case study of a drug that has a half-life of 72 hours. What study design would you choose for a relative bioavailability study? A crossover or a parallel study design? Generally, I would prefer a crossover design, and parallel should be considered when crossover design is not feasible. In a bioavailability study or a PK study involving an immediate release oral product with a long half-life, a characterization of product's half-life should include blood sampling over an adequate period of time. To determine the bioavailability of a drug product containing a drug with long half-life, a single-dose crossover study should be conducted if an adequate washout period is used. If the crossover study is problematic, a study with parallel design should be used. Ensure that the similar subject demographics in each treatment groups. And for either a crossover or parallel study, the sample collection time should ensure that the drug product completely moves through the gastrointestinal tract so that the complete absorption of the drug substance can be obtained. A suitably truncated AUC can be used to characterize the peak and total drug exposures, and this can be done for drugs that do not exhibit flip-flop kinetics and drugs that do not have high intrasubject variability. This slide shows an example of truncated AUC for a drug that is slowly eliminated with a half-life for about a week. A profile is shown in the right, and for this product, AUC truncated at 48 hours or 72 hours has been used for comparing bioavailability. Now I want to discuss highly variable drugs and narrow therapeutic index drugs. High variability drugs and drug product are defined as those having high intra-subject variability of greater than or equal to 30%. This is the variation in the same subject taking the same drug in two different occasions under similar conditions. And these drugs generally exhibit a wide therapeutic window. For such products, a reference-scaled approach using a replicate design where subjects receive the reference product more than one occasion can be considered. And this approach reduces the sample size required for the study. Narrow therapeutic index drugs are those drugs where small differences in dose or blood concentrations may lead to serious therapeutic failures and/or serious adverse drug reactions. Some examples are listed in the slide: warfarin, digoxin, etc. For the narrow therapeutic index drugs, knowledge of exposure measure of drugs like AUC or Cmax are critical for the safe and effective use of the drug product. And also, therapeutic drug monitoring is an essential tool for drug product dosing. Therefore, for such drug products, tighter limits for bioavailability assessment and product quality standards are required, and the acceptable criteria for demonstrating the bioavailability might need to be narrowed. The guidance includes the reference cited below, and you can refer to this for more details. But usually in this situation, bioavailability determinations are done using a scaling approach, using a four-way, fully replicated crossover design in healthy subjects. And this allows simultaneous comparison of the mean and within subject variability of the test and reference product. And because both test and reference product are given twice, the four-way crossover replicate study design enables the scaling of the acceptable limit to the within subject variability of the reference product. And the proposed limits of the NTI drugs of 90% to 111.11% can be scaled based on the within subject variability of the reference product. So let's talk about a case study of no pain, which has a greater than 50% intra-subject variability. What approach is appropriate for a relative bioavailability study? As this product has greater than 30% intra-subject variability, we can consider a reference scaled approach to reduce, which may reduce the sample size needed for the bioavailability assessment. With that, I would like to conclude my talk and briefly summarize what we discussed here. We discussed some special topics and approaches to determine bioavailability. For example, the baseline correction for endogenous compounds, bioavailability of individual components in an fixed dose combination. I also presented when we can choose a reference scale study design and considerations for long half-life drugs and narrow therapeutic index drugs. As all these special topics have various complexities involved, sponsors should consult the appropriate review division to discuss their specific situation. With that, I would like to thank you for your attention. Please submit your questions. I'll come back in the panel discussions. Thank you.
Dr. Kumi.
Good afternoon. I hope you had a good break. My name is Kofi Kumi. I'll be discussing pharmacodynamic and non-traditional endpoints used in assessing relative bioavailability. This is my disclaimer. The objective of my presentation are as follows. First, I'll talk about non-traditional endpoints concentrating on partial AUCs and point-to-point concentration comparison. I'll then discuss pharmacodynamic endpoints using pulmonary products and topical product as examples. Then I'll talk about clinical endpoints using topical product as examples. What are the approaches that are recommended to evaluate bioavailability? Peak endpoints are preferred because they are the most accurate, sensitive, and reproducible. Pharmacodynamic endpoints can be used if peak endpoint is not possible. The PD endpoint should be well justified. Other approaches to evaluate viability include in vitro studies, for example, in vitro dissolution, where the similarity factor comparison is used as an endpoint. Comparative clinical studies, when measurement in an accessible media is not possible, or PD is not possible. Please remember this is the least sensitive approach. Now let's talk about non-traditional PK concentrating on partial AUCs. Certain class of drugs, Cmax and total AUC, may not be sufficient. Onset and duration for these class of drugs are critical considerations. Examples include complex dosage form for ADHD drugs and analgesics. Time to truncate partial AUC should be related to a clinically relevant exposure measure. Sufficient quantifiable samples should be collected to allow adequate estimation of the partial AUC. It's always advisable to consult the review division. Let's look at some examples. Example 1 is a new extended release formulation of drug A. Drug A is indicated for the treatment of attention deficit hyperactivity disorder or ADHD in adults, adolescents, and children. The desired exposure profile is coverage during waking hours for patients to be productive at school or work but able to sleep at night. Product A is an immediate release dosage form of drug A. Product B is an extended release dosage formulation of drug A. Product B is intended to provide coverage up to 12 hours. However, physicians commonly prescribe an additional dose of product A after 8 hours of product B due to possible lack of effectiveness for the 12-hour intended duration. So for this program, the reference clinical regimen was 1 dose of product B in the morning and 1 dose of product A 8 hours later. The sponsor developed product C, which is a once daily triple bead sustained release formulation. The difference between product B and product C is that product B contains 2 different beads, product C contains 3 different beads. The third bead in product C provides additional relay release that is intended to provide symptomatic relief for up to 16 hours. The application was submitted via the 505(b)(2) pathway. This is plasma concentration time profile of the BA results comparing product C to product B plus product A, the clinical regimen. The filled circles represent the product C profile, and the open diamonds represent product B plus product A profile. That is the profile for the clinical regimen. Please note, Tmax for product C is about 8 hours. Tmax for product A plus B, the reference regimen, is about 10 hours. Tmax for product B is about 5 hours. 12 hours is the duration of duration of school or work hours. And the sponsor is claiming that this product C is effective for up to 16 hours. So what PK parameters would you recommend to the sponsor of product C to evaluate and determine whether the 2 dosing regimens are equivalent? The study will be conducted under both fed and fasting conditions in adults. By equivalence, PK parameters recommended will be Cmax AUC 0 to T or 0 to last and AUC 0 to infinity. Therapeutic equivalence will be AUC 0 to 5 hours and AUC 5 to 12 hours. This is a summary of the rationale for the PK parameters that would be recommended or were recommended. By equivalence, Cmax, AUC 0 to last, and AUC 0 to infinity. AUC 0 to 5 is for the early onset of response. AUC 5 to 12 is for sustaining the response based on school work hours and Tmax of the sustained release formulations. AUC 12 to 16 will be for maintenance of response based on an efficacy claim. Please note that because of this efficacy claim, the sponsor did conduct a safety and efficacy study. Product Y is an extended release formulation of drug Y, which is approved as an immediate release formulation. Drug Y is indicated for the treatment of epilepsy. No safety or an efficacy study was conducted in this program. This application was submitted via the 505(b)(2) pathway. The listed drug was the immediate release dosage form of product Y. PK exposure comparisons were AUC, Cmax, and Cmin. Cmin because exposure response analysis have indicated that reduction of seizures is correlated to semen. Point-to-point concentration comparisons were done, and then partial AUC comparison also were evaluated. This is a plasma concentration time profile of comparison between the extended release formulation versus the IR formulation of drug Y. The red line represents the profile for the extended release formulation. The blue line represents the profile for the immediate release formulation given every 12 hours. This is a summary of the statistical analysis for relative bioavailability of 200 milligram dose of the extended release formulation and the immediate release formulation given at steady state. As you can see, the mean ratio for AUC were fairly comparable, for semen very comparable, and for CMAS was okay, but as to be expected, the CMAS for the extended release formulation is lower than that for the immediate release formulation. And as you can see from the 91st confidence interval are all contained within the 80 to 125 regulatory criteria for bioequivalence. This is a point-to-point comparison of the concentration at each time point. For drug Y, 200 milligram QD of the extended release formulation, and 100 milligram every 12 hours for the immediate release formulation. The squares are the profile for the extended release formulation, and the circles are the profile for the immediate release formulation given every 12 hours. This is analysis of the point-to-point drug Y concentrations after XR and IR administration, and for each time point from 0 to 24, as you can see, the ratios were fairly comparable, and the 90% confidence interval contained within the 80 to 125 regulatory criteria for bioequivalent, except at the early time points of 0.5, 1 hour, and 1.5, where the lower limits were outside the 80% lower limit of the 80 to 125 regulatory criteria. This is point-to-point comparison of the partial AUCs and the ratio of the concentration at each time point. As you can see, the 90% confidence interval of the partial AUCs at each time point were all contained within the 80 to 125. For concentration at each time point, they were all contained within the 80 to 125, except for the early time points, as I indicated earlier, where they were outside the 80% lower limit for bioequivalence. So based on this analysis, this product YXI was approved based on point-to-point concentration comparison, partial AUC comparisons, and traditional bioequivalent comparisons. So no clinical studies were conducted for this program. Now let's discuss pharmacodynamic approaches that can be used to assess bioequivalence and or bioavailability. PD endpoints with sufficient accuracy, sensitivity, and reproducibility can be used to assess bioavailability and or bioequivalence. The biomarker selected should be relevant to the disease process, Dose-response relationships should be demonstrated, and always advisable to consult with the review division. Let's look at some high-level examples of PD approaches to demonstrate bioavailability and/or bioequivalence. Arbitrary metered-dose inhaler, where forced expiratory volume is used as the PD endpoint. For other inhalers, for example, inhaled corticosteroids, where exhaled nitric oxide is used as the PD endpoint. Topical ointments and creams, for example, fluticasone propionate, where skin blanching is used as a PD endpoint. I've provided some references where additional information can be obtained for these PD endpoints that have been used to assess bioavailability and/or bioequivalence. Now let's talk about topical products, how to assess bioavailability for topical products. Topical delivery systems represent formulations that are intended to deliver drugs locally rather than systemically to treat local pathophysiological conditions. Clinical study may be considered sufficient for measuring bioavailability or demonstrating bioequivalence of dosage forms intended to deliver the active moiety locally. For example, topical preparation for the skin, eye, and mucous membranes. Some recommendations for conducting clinical trials for topical products. The trial itself could be a standalone trial in phase 2, or could be a subgroup of subjects in a larger phase 3 trial. Either approach is reasonable. The sponsor should demonstrate that systemic exposure from their product and the maximum usage condition is comparable to a comparator product, so that systemic safety will also be comparable. If there's no comparator product, the systemic exposure should be evaluated in light of systemic adverse events. So it's advisable to consult the review division when planning clinical trials as to evaluate bioavailability and bioequivalence. Additional requirements for topical products. Evaluation of the vasoconstriction activity of topically delivered corticosteroid in normal skin and healthy adult subjects can be the basis of potency ranking and BE of corticosteroids. Agents having the potential to suppress hypothalamus pituitary adrenal should be tested for possible HPA axis suppression. So in summary, non-traditional PK parameters may be needed for some disease conditions and complex drug products. For example, analgesics, complex ADHD drugs. Onset of duration, important consideration for this type of products. Partial HC's point-to-point concentration comparison may be reasonable under certain scenarios. PDL is reasonable when PK is not feasible. Clinical studies allowed if PK or PD not possible. Example for topicals. And always it's advisable to consult with the review division so that both the sponsor and the agency have a common understanding of what is to be expected for the program. I would like to acknowledge these people for their comments and input in the presentation. My supervisors, Mehu and Ramana, Tachen and Angela, evaluate the NDA for drug Y, the point-to-point concentration comparison NDA. Jordan Lau provided the references for the PD endpoints. Tapash provided the slides and input to the studies for the topical drugs. Dakshina, Jaya, Oppo, Ethan, Paul, members of the planning committee for this webinar for their input and comments for the presentation slides. And the members of the BA working group for drafting and finalizing the BA guidance for which we are here discussing today. Thank you very much for your attention. And I believe question and answer section will be during the panel discussion.
Good afternoon. My name is Okpo Eraderi. I am one of the branch chiefs in the Division of Biopharmaceutics. I'm really excited to talk to you today about recommended in vitro studies in the guidance. My excitement is partly due to the fact that if what we are going to go through today in this presentation, if those principles are applied appropriately, you are going to be able to save R&D costs for your company. And I believe it is generally accepted that in vivo studies that are very simplistic are usually more expensive than the most complex in vitro studies. This is a disclaimer that you've seen a number of times already today, and I just want to mention that this applies also to my my office is also located, my division is located in the Office of Pharmaceutical Quality, and in that office, extreme care is taken in assessing the quality of drug products to ensure that patients continue to consistently get high quality drug products throughout the life cycle of the shelf life of the drug. First of all, I want to talk about the in vitro approaches for bioavailability in general in this guidance. In general, a batch release test dissolution can be used to measure bioavailability. And in this particular guidance, we have several references that are made to a number of guidances, other guidances, that talks about the method development, method optimization, and method validation for dissolution. The second approach is regarding extended release drug products. These dosage forms are such that if a dissolution method is developed to have predictive characteristics, in other words, if in fact the dissolution method can be biopredictive, then an in vitro in vivo correlation model can in fact be established. If this model is assessed and deemed acceptable by the FDA, then post-approval changes on that particular product and that particular formulation can in fact be approved without the need for in vivo bioavailability studies. And this can be in perpetuity, provided that no change is made to the formulation itself. Now, today I'll be covering biowaivers, which is the cornerstone of this particular topic. And thereafter, we'll go ahead and talk about recommended in vitro studies for immediate release products, which are oral and immediate release products. Thereafter, we'll go ahead and talk about the modified release products, other modified release products, that is, modified products that are not tablets, capsules, or suspensions. And then a very important subtopic, alcohol dose dumping, will also be covered. And at the end, I'll give you a summary of the talk itself and a couple of take-home messages within the summary. Now, as I indicated, biowaivers are very important. The requirements for biowaivers are stipulated in 21 CFR 320.22, and if granted, if the biowaiver is granted, then no additional bioavailability or bioequivalence studies are needed. Based on the in vitro data, usually, of course, the dissolution data, in vivo data requirement is waived. In vivo data requirements are usually waived. And also, if this waiving of biostudies are usually for lower strengths, because in many cases bioavailability studies are done on the higher strength. But in scenarios where, in cases where you have the higher strength for which you need a biowaiver to be granted, then linear pharmacokinetics will usually be required. Now, for multiple strengths, there must be proportional similarity, and that is all of the active and inactive ingredients are in identical proportions between the different strengths. For high potency drugs, you can also achieve proportionality, but I consider this to be some kind of a carve-out for high potency drugs. In such drug products, the drug composition, the drug itself makes up less than 5% of the total composition of the total weight of the tablet or the capsule contents. And the other component, the other aspect that I'd like to also highlight are bilayer tablets. Bilayer tablets are considered a single formulation, even though they consist of two separate layers with different compositions. All components of both layers should be proportionally similar. Now, what if the different strengths are not proportionally similar? What do you do? I did mention at the beginning of this talk that we are here to give you hints on tips on how you can save around the costs. There is also a caveat for when you don't have proportionality between strengths. And one of the ways that you can do that is to, again, use the biowaiver, the dissolution testing, comparative dissolution testing procedure. And as long as the ratios of the inactive ingredients to the total weight of the dosage form are within the limits defined by FDA's SUPAC-IR and SUPAC-MR guidances, then it will be deemed, the strengths will be deemed to be proportional if in fact they meet the requirements for SUPAC-IR and SUPAC-MR, and the changes, the differences of the changes are within the SUPAC limits. Then you can proportionality will be assumed. The other important aspect also is that for fixed combination products, it is unusual to see proportionality between strengths, because these fixed combination products comprise of more than a single active entity, and one of the components is usually fixed in composition, while the other one is modified or changed. And for this type of products, we expect that a biowaiver is possible for the intervening strengths. In other words, if the in vitro dissolution is such that you can demonstrate similarity, then you can actually you'll be able to request a biowaiver for the intervening strengths, but not for the highest and lowest strengths, because these are combination products that are not likely to be proportional. At this point, I'd like us to transition to the various types of in vitro studies that are conducted in support of bioavailability. In the Code of Federal Regulations, 21 CFR 320.24, FDA is given the authority to determine that an in vitro approach is appropriate to determine bioavailability or bioequivalence. I will now describe the guidances, recommendations on such studies for oral immediate release and modified release products. As I indicated before, in vitro dissolution data are needed to measure the bioavailability of capsules, tablets, and suspensions. Now, if the various strengths of a particular drug product are pH independent, then there is a much more simplistic approach that can be taken to do the in vitro, comparative in vitro dissolution and to generate that data for different strengths. If there's pH independence in the drug release rate, then just doing the dissolution test with the regulatory method that is proposed is all that is sufficient. However, if there is pH dependent or strength dependent dissolution for the drug product, then the comparative dissolution testing will need to be performed at 3 pH values, 1.2, 4.5, and 6.8. pH values that are considered to be the physiologic pH range. And one of the way to determine whether in fact the in vitro dissolution between strengths are similar is because it's to do a test, to use the test statistic, the F2 test. And once the F2 is greater than 50, then the dissolution data is said to be similar across strengths. Now, I'd like to talk about over-encapsulation. As you all know, when a clinical trial is about to be done, in order to maintain blinding in the clinical trial, drug products are usually over-encapsulated. And the over-encapsulation is such that we do not expect any changes in the drug release profile. One of the requirements of over-encapsulation is that the over-encapsulation process itself is not affecting drug release in the in vitro dissolution test. If a new excipient also is added during the over-encapsulation process, then an in vivo study will be required. Comparative dissolution, if and when demonstrated, would be such that there will be no need for an additional in vivo study. The key is to demonstrate and to prove that there is no impact on drug release. The other point that I'd like to make is the fact that enzymes can be used in the comparative dissolution testing in this regard. Because the enzymes, the capsule shells can slow down the rate of drug release. So enzymes can be added to the dissolution medium to add to the dissolution medium to remove the potential impact of slowing down of the drug release because of the capsule shells. And then in terms of the SUPAC IR, which is the scale-up and post-approval changes, we do have a guidance that lays out all of the details as to what the requirements are for SUPAC IR for post-approval changes. And these changes can either be formulation changes or they are manufacturing changes. Now, formulation changes can occur in components and composition, while manufacturing changes are quite a bit, potential manufacturing changes can be the manufacturing site, it can be the manufacturing process itself, or it could be the batch size. Batch sizes usually are increased, but in a few cases, in fact, just a few days ago, I've come across, we have just received a supplement that proposes to reduce the batch size, which will then lead to the change in equipment as well, for the smaller batch size. So all these, regardless of all these changes, are the responsibility of FDA is to make sure that the release rate, the drug release rate, hasn't actually changed. Now, I talked about various types of changes. Based on these types of changes, there are generally 3 levels of change. Level 1, Level 2, and Level 3 changes. Levels 1 and 2 are considered minor changes, but Level 3 changes are usually considered to be major. And for immediate release products, Level 3 changes for immediate release products usually will require dissolution testing in several dissolution media, whereas Levels 1 and 2, you may use only the regulatory method to demonstrate that in fact there has been no changes. So there is the SUPAC IR guidance itself. We can probably address more details if necessary during the Q&A session, or you may refer to the SUPAC IR guidance. Now going to the SUPAC MR guidance, the similar requirements also are there, and dissolution data can be used to support the changes. And again, for details, I would recommend the SUPAC MR guidance itself. Now, for beaded capsules that are modified release or extended release, these beaded capsules usually are made in such a way that the strengths differ only in fill weight. And sometimes these beaded capsules are referred to as multi-particulate systems, and because they differ only in the fill weight, proportional similarity is quite evident in this particular case. Now, if the strengths for which a biowaiver is needed, or or a particular change is needed, or a biowaiver is needed, then usually they're the lower strengths. Now, if a higher, and I think I mentioned this earlier on in the talk, that if you are planning to submit a biowaiver request for a higher strength or for higher strengths, then in addition to the comparable dissolution profiles, you will need to ensure that the pharmacokinetics of the drug are linear. And if the dissolution method has actually not been finalized, because usually you just use your regulatory dissolution method for all of these testings, but if the dissolution method has not been finalized during drug development, and you still have the method as being putative, then our recommendation, the guidance recommends that you do your testing at pH 1.2, 4.5, and 6.8. Now, for other modified release dosage forms that are not capsules, tablets, or suspensions, as has been stated before, in vivo bioavailability study is usually conducted on the higher strength, and the lower strengths, BA studies on the lower strengths can in fact be waived. And the conditions are similar to what has been mentioned in previous slides, and that is that the dosage form itself is the same between the lower strengths and the higher strength on which bioavailability data has been generated. You demonstrate proportional similarity, and the same mechanism of drug release is present in all of the strengths, and also linear pharmacokinetics. As is usually the case, there are usually differences, or you may come across an issue where you've done all of your clinical trials and you've demonstrated safety and efficacy, and towards the latter end of your R&D cycle, you find that all of a sudden from the clinical pharmacology area, that department, that in fact the strengths are not proportionally similar. So if the strengths are not proportionally similar, then what do you do? And it's very simple. If you have more than two strengths, you'll be able to do the bioavailability, generate the bioavailability data on the highest and lowest strengths, and you'll then be able to request a biowaiver for the intervening strengths. And again, the same kind of requirements, that is, you demonstrate similar multimedia dissolution profiles. Similarity factor F2 is greater than 50, but for these modified release dosage forms, you can actually develop a dissolution safe space via IVIVC or IVIVR. And you may have also, you can also have virtual B studies conducted in addition to these models, and be able to use that to support and to justify the biowaiver request. We're now going to the last section of this presentation, which is the alcohol-induced dose dumping. We all know about the potential of alcohol-induced dose dumping from modified release dosage forms. If in fact you have an extended release dosage form that contains the dose of an entire day in one unit, and if for some reason that dose is released all at once, then you're going to have some exaggerated pharmacological effects which are undesirable. So in the labeling of many products, I think many drug products, there are instructions there not to take the drug with alcohol because of these pharmacodynamic effects that may be exacerbated or caused by the drug-alcohol interaction. For modified release products, however, you can have a little bit more complexity. The alcohol may in fact cause the entire dose to be released very rapidly, and that is what we term as a biowaiver. I'm sorry, the dose dumping, and also the undesired pharmacological effects as well, which we do not want. But again, the good news is that the risk mitigation is quite simple. You don't necessarily have to go and conduct a clinical trial by co-administering the drug with alcohol. You don't have to do even bioavailability studies. The in vitro studies that I've been talking about, again, can be used to mitigate the risk of alcohol dose dumping. What this guidance recommends is that for alcohol dose dumping, you do dissolution studies on the highest and the lowest strengths. The optimum dissolution method should be used for such studies, and the alcohol concentrations that are recommended are 5, 20, and 40%. Obviously, the medium that contains 0% alcohol would be the medium in the regulatory dissolution method. A sample size of 12 is recommended in the dissolution test. In other words, you have to use 12 units of the dosage form. And also, we strongly recommend multiple time points beyond, especially within the first 2 hours and beyond, so that we can obtain the full dissolution profiles. Now, talking about the dissolution medium itself for alcohol dose dumping study, in vitro alcohol dose dumping studies, the dissolution medium of choice is 0.1 normal HCl. That is the default medium, and with the appropriate apparatus and agitation speed. If 0.1 normal HCl is the medium in the proposed dissolution method, in vitro tests with the various alcohol concentrations previously mentioned will be sufficient, and that completes the test. If in fact 0.1 normal HCl is not the dissolution medium that is being proposed for use in the regulatory method, then you need to generate dissolution profiles using the recommended alcohol concentrations in 0.1 normal HCl as above, as previously described, and in the optimum dissolution medium itself. So you actually run slightly longer experiments. But the key is to make sure that at the end of the experiment, the main question to be answered usually is whether the modified release characteristics are maintained, and then to estimate the F2 similarity factor for each alcohol concentration versus the control, which is the medium without alcohol. And we always ask for individual unit data to be provided, to be tabulated, and also plots and descriptive statistics. One of the, again, good things about this guidance is that this idea of investigating alcohol dose dumping is that the results are very simple. You either demonstrate dose dumping or you don't, or there is no dose dumping. If there is no dose dumping, your experiment is over. You've done the appropriate study, and you've completed this particular type of experimental data. But if there is dose dumping, what happens? If there is dose dumping, do you go ahead and run an in vivo study, in vivo bioavailability study? Or do you try to capture these results in the labeling? Do you have other risk mitigation strategies? And that's when we recommend that, in fact, you contact the reviewing division to discuss this and to determine the best course of action prior to submitting your application to the agency. Now I'll summarize. Bioavailability and occasionally bioequivalence can be assessed with in vitro studies. In vitro data can be surrogates for bioavailability and sometimes bioequivalence. These principles that I've described and indicated that they are actually in guidances specific for immediate release products and guidance specific for modified release products. They are actually post-approval requirements, but these changes can occur even pre-approval. And so the principles that are the principles within the super guidances are applicable also to pre-approval situations. And the type of dosage forms, whether it's immediate release or modified release, you can actually use the in vitro dissolution test to measure bioavailability. In addition, we talked about multiple dosage strengths, that in fact these concepts, these principles are applicable to multiple dosage strengths. Also, that alcohol can have an impact on drug release and absorption from modified release products. And if you demonstrate absence of dose dumping in your experimental work, then you don't have to do anything further. Your experiments are complete. If, however, you demonstrate dose dumping, well, the guidance recommends that you contact the FDA reviewing division before filing. And I'd like to acknowledge Kofi, Dakshina, also Bhagwant, and Ramesh for their contributions towards the preparation of this presentation. Thank you very much, and I'll be back with the rest of the panel, with the rest of the speakers for the panel discussion and the Q&A. Thank you.
Thank you all for the great presentations. We'll now enter into our Q&A panel discussion. As a general reminder, if you haven't had a chance to enter your questions into the Q&A chat pod, please do so now. We'll answer as many questions as time allows. To start us off, we're going to welcome to the panel Dr. Ethan Stier. It looks like we have about 4 questions that were sent for Dr. Stier. And here is the first question. Does a comparative BA study have to meet BE criteria?
Thank you. There is no regulatory requirement for a comparative study to meet BE criteria. The BA guidance outlines if a study does not meet the 90% criteria for Cmax or AUC, then the sponsor will need to demonstrate that those differences don't affect safety or efficacy. In some cases, the firm may need to consider differences in Tmax or differences in the shape of the curve, which we refer to as partial AUCs. And also, in some cases, strict bioequivalence criteria may be applied because of the pharmacology of the drug, for example, narrow therapeutic index drug, which is also covered in the guidance. And of course, in general, even though there's not a formal bioequivalence requirement, with some exceptions, as I mentioned, the study should be properly designed to provide confidence in the estimate of the relevant PK parameters. And of course, a lot of the things I've talked about are unique to the pharmacology of the drug and the dosage form of the drug product, and really should be addressed to the review division during the IND discussions. Thank you.
Thank you for responding to that question, Dr. Stier. We do have a few more questions for you, and here is the next question. Where a food effect study of a drug designed for a once daily dosing demonstrates an effect of food on the rate and extent of absorption, are instructions to take on an empty stomach at least 2 hours before or 1 hour after food appropriate for labeling for this drug?
Thanks. For that question, that's outside of the scope of the guidance that we talked about today. And I would refer the participant asking that question to go to the food effects guidance called Assessing the Effects of Food on Drugs and INDs and NDAs that was published in June of this year.
Thank you for responding to that question. We got a couple more questions for Dr. Stier, and here is the next question. What are the differences between relative BA study and bioequivalence studies?
Sure. So I think I talked, in my first question that I received about the relative or comparative bioavailability study. So bioequivalent studies, as I believe it's mentioned here in the question, or is intended in the question, that's really referring to what is used by the Office of Generic Drugs to evaluate generic drug applications, also called abbreviated new drug applications. And also in the introductory slides, I provided a link to that guidance, the PK Endpoints guidance. And I also believe there's an SBI webinar that was recorded earlier this year that also discusses that guidance in detail. So I think for information about that, the questioner could seek that information out there.
Thank you for responding to that question. We do have one more question for Dr. Stier, and here is the question. The terminology combination product used here may be confused with combination products used under the Office of Combination Products where they are a drug device, drug biologics. How can the terminology be distinguished?
That's a good question. So just to be clear, I guess for today's presentation, when we were talking about drug combination products, we're really talking about two different drugs or two different APIs that are co-packaged together or formulated together as a fixed dose combination product. And that's true. There is somewhat similar terminology referring to drug devices, and that's also handled by other centers, which is kind of the outside of the scope of today's presentation. So I'll just kind of leave it at trying to define what we were talking about today during the presentation.
Thank you, Dr. Stier, for responding to those questions. We're moving on to our next panelist. We do have a few questions that came in for Dr. Jayabharathi Vaidyanathan. And here is the first question. Please outline under what circumstances the agency would find uncorrected AUC acceptable for a biological endogenous compound that is difficult to measure or a treatment that is unsafe to interrupt for washout.
Hi, I would answer this in separate this question into three different parts. First, under what circumstances would we find uncorrected AUC acceptable for endogenous compound that is difficult to measure? So generally, as per the guidance, we recommend that the sponsor present both the adjusted and unadjusted PK parameters for bioavailability assessment. And for example, if that endogenous compound is difficult to measure, on occasion we have used a much higher dose than what is given therapeutically, provided it is safe to administer in order to overcome that bioanalysis difficulty. So if, and please contact the division for case by case when it may be acceptable to just use uncorrected. And on the other question, when it is difficult, unsafe to wash out, generally we recommend BA studies to be conducted in healthy subjects, especially to avoid such issues when conducted in a patient. So please reach out to the division for specific cases. Thank you.
Thank you for responding to that question, Dr. Vaidyanathan. And you got a little bit of an echo in your volume. So what we're going to do is give our tech team an opportunity to reach out with you. And we're going to move back up to Dr. Dakshina Chilukuri for a few questions, then check back in with you after we've asked a few questions with Dr. Chilukuri. And here is the first question. Excluding a gender without a specific reason would that be a reason to deny the dossier?
Yeah, thank you for that question. In general, in the IND and NDA world, there is no specific denial of a dossier per se, but typically the guidance recommends that both subjects of both genders be included in the relative BA studies. So if for some reason a specific gender is not going to be, or if a sponsor would like to not include a specific gender, then an explanation or some sort of a rationale provided would be useful. And a discussion with the specific therapeutic area or the division could be made to support that particular point of view. Thank you.
Thank you for responding to that question. We have a couple more questions for Dr. Chilukuri, and here is the next question. What are the recommendations regarding sample size for RBA studies?
So the relative bioavailability studies, the sample size recommendations depend on the PK properties of the drug, depending on the variability in the PK for these drugs. So no specific recommendations per se, but the most commonly used number vary anywhere from between 12 and 24 subjects per arm, depending on the variability of the drug. If it's a highly variable, if the drug is characterized by high variability, then essentially a different number, a higher number could potentially be used. So no specific recommendations, but it would depend on the PK properties of the drug. Thank you.
Thank you for responding to that question. We do have a couple more questions for Dr. Chilukuri, and here is the next question. It's regarding single-dose studies with linear PK. Are they sufficient on the highest strength if no analytical concern, and there's not necessarily a need to conduct the studies on the highest dose? Is this a correct thinking?
Thank you. That is correct. For single dose studies with linear PK, if no analytical concern, then yes, the study could be conducted on the highest strength without the need to conduct a study with highest dose.
Thank you for responding to that question. We do have one more question for Dr. Chilukuri, and here is the question. Considering the challenges for recruitment of female volunteers, can only male volunteers be included in a BA study? And is there any justification required to be documented in the report for this same issue?
Question that was asked earlier. So the recommendation is to provide some justification for selection of one gender over the other, or not having both subjects of both genders included in the BA study. So some sort of justification and any other data that the sponsors have at hand would be useful in that would be useful in making a determination whether this would be acceptable or not. So yes, some sort of justification would be helpful, either previously obtained PK data or any other justification that the sponsor could think of. Thank you.
Thank you for responding to that question. We do have a few more questions for one of our next panelists, Dr. Kofi Kumi. And here is the first question for Dr. Kumi. It's regarding slide number 13. On slide number 13, were both the IR and ER tablets BAB results comparable?
Thank you for that question. I'm assuming slide 13 that you're referring to is for drug Y. And if you can look on slide 14, following that, it does show that they are comparable, comparing the XR2 are comparable based on the 90% confidence interval.
Thank you for responding to that question. We do have a little bit of static coming from your microphone, Dr. Kumi, so the tech team is going to reach out with you and troubleshoot that for a moment. And we're going to move back up to Dr. Vaidyanathan for a couple more questions to see if the volume comes through. And here is the first question for Dr. Vaidyanathan. How does one apply the evaluation of pre-dose concentration being greater than 5% of Cmax if the drug of interest has endogenous levels?
Hello. Hopefully the audio is much better now. So in general, for endogenous compounds, again, it's very important to measure the pre-dose concentrations. And in this case, I'm not clear why concentrations are higher than or greater than 5% of Cmax, but I would recommend multiple time points for pre-dose measurement of concentrations and subtract it from post-dose. If there are still questions, I would recommend contacting the review division for your specific case. Thank you.
Thank you for responding to that question. You're coming in good and clear. We're moving on to the next question for Dr. Vaidyanathan. And here is the next question. How do you define comparable bioavailability? And it's in slide 9, they think.
The example that I had used in slide 9, it was for a combination product. And what I meant by comparable bioavailability was in that particular example, the two moieties, the two drug coming from the fixed combination product met the criteria for the 90% confidence interval, and it was within the 80 to 125%. So that was what I meant by demonstrating comparable bioavailability to the individual reference listed products. Thank you.
Thank you for responding to that question. We've got a couple more questions for Dr. Vaidyanathan. And here is the next question. In case study 2, as you mentioned the desirable study design of FDC versus A plus B, knowing the existence of the interaction between drug A and drug B, does the study need to include clinical endpoints as well?
This specific example, we are only talking about PK determination of bioavailability. And even though there is a drug interaction between the two drugs, it is not needed to have a clinical endpoint in this case because the drug interaction between the two arms would be the same that you would expect when you give in such a design, that is evaluating the fixed dose combination with the two drugs given concurrently. So just understanding the PK in this situation would give us a good understanding of the relative bioavailability. Thank you.
Thank you for responding to that question. We do have one more question for Dr. Vaidyanathan, and here is the final question in this round. How do we determine the number of pre-dose time points required for baseline correction?
I think it's a good question. Generally, what I have seen is at least 3 time points for pre-dose measurements for baseline adjustment. And again, it depends on the drug, but at least on many of the cases, we have seen at least 3 time points pre-dose. Thanks.
Thank you for responding to those questions. Moving on to our next panelist. We do have a few questions that just came in for Dr. Akpo Aradiri. And here is the first question for Dr. Aradiri. What would be the recommendation for a situation where the kinetics are linear but not proportional? Will this be single strength or high and low?
I'm sorry for the pause. Yeah, great question. If in fact the kinetics are linear, but your strengths are not proportionally similar, then we would go ahead, and I think what my recommendation would be is for you to do the bioavailability studies on the highest and the lowest strengths, And if you have intervening strengths, I think it would be appropriate then for you to be able to, you can then request a biowaiver for the intervening strengths, similar to what you would do, for example, if the kinetics were non-linear. Thank you.
Thank you for responding to that question. I have a couple more questions for Dr. Aradiri, and here is the next question. If the drug product is labeled to be taken with food, would this mean that the IV-IVC study should be conducted under fed conditions?
This is rare, actually. It's not very common to see IV-IVCs being done with in vivo data generated in the fed state. But in this particular situation, a hypothetical situation where you are having where the drug product is supposed to be administered only with food, then if you do the classical IV-IVC in the fasting state, first of all, you'll be violating the label if you administer the drug. So from a clinical perspective, I think it's appropriate that you would make an attempt to establish an IV-IVC with in vivo data generated from a fed study. Thank you.
Thank you for responding to that question, Dr. Aradiri. We do have a couple more for you. And here is the next question. Regarding pre-approval stage, if pH 3 formulation is a common blend and has linear pK when compared to the to-be marketed formulation, can a BA study be done at a lower strength, and should we ask for a waiver for the higher strength of the to-be marketed formulation?
I have a little bit of a problem here. Can you please repeat the question, the first part of the question?
Yes, and we'll repeat the question. And it's the first part of the question references the pre-approval stage. If pH 3 formulation is a common blend and has linear pK when compared to the to-be marketed formulation with BA study, can BA study be done at a lower strength and ask for a waiver for the higher strength of the to-be marketed formulation?
Yeah, thank you very much for repeating the question. I think normally we would prefer that the higher strength or the highest strength be used in the PK study, and you request biowaivers for the lower strengths. However, if there is a potential problem of toxicity, or perhaps if you are doing the study in healthy volunteers and they cannot tolerate the dose equivalent to the highest strength, in such a situation, it would be appropriate for you to do the study on the lower strength and ask for a biowaiver for the higher strength. In fact, I would even go further to say that you don't necessarily have to use the lowest strength per se. You can simply use one of the lower strengths, and then you can ask for a waiver for the higher strength, and also for any strength that maybe if there are other lower strengths, you can ask for a waiver for those as well. Hopefully that is clear. Thank you.
Thank you for responding to that question. Moving up to the beginning of our panel, we do have another question that came in for Dr. Ethan Stier. And here is the question that we have right now for Dr. Stier. What happens for post-approval if the BE is 80 to 125% if is not met in the BA study? What additional studies are required?
Yes, in that case, I would encourage the applicant to reach out to the review division to discuss the particulars of that situation.
Thank you for responding to that question. Moving on to our next panelist, we've got a few more questions that came in for Dr. Chilukuri. And here is the next question, or the first question for Dr. Chilukuri. When should a multiple dose BA study be conducted?
Sure. So most of the times, what we see are the single dose studies, single dose BA studies, BA studies conducted under single dose conditions, mainly because of the sensitivity that they are considered to be the most sensitive. In some cases where, for analytical reasons, it is conceivable that multiple dose BA studies could be conducted. And again, we encourage sponsors to reach out to the specific therapeutic area or the division to discuss further. In addition to that, I think I've covered this under the multiple dose MR studies slides where I talked about scenarios where the multiple dose modified release formulations for which the multiple dose studies are usually conducted to document the steady state pharmacokinetics of the drug. So these are the scenarios. Thank you.
Thank you for responding, Dr. Chilukuri. We do have a couple more questions for you, and here is the next question. Rather than conducting two separate single-dose fasting and food effect studies, is it usually acceptable to conduct a single three-arm study, test fast, RLD fast, and test fed?
Yes. So the guidance recommends, as it was described in the Appendix B of the guidance, and the way I've talked about it during the presentation, there is no specific recommendation for conducting two separate studies versus a three-way crossover study. It is actually up to the sponsor to decide. Considerations regarding the cost of conducting a three-way crossover study versus two separate studies is something that we have seen. So that could be one consideration, but and also getting the results in one study may be preferable for some sponsors. But to answer the question, no, there is no specific preference. It's up to the sponsors how they want to approach this.
Thank you for responding to that question. We have one more question for Dr. Chilukuri, and here is the question. Is there a batch size requirement similar to a BE study for the RBA study?
So this guidance does not talk about batch size requirements, and so the answer is no. There are no specific batch size requirements for the RBA study. Thank you.
Thank you for responding to that question. Moving on to our next panelist, we do have a few more questions that came in for Dr. Jayabharathi Vaidyanathan, and here is the first question for Dr. Vaidyanathan. How can a drug be classified as indexed narrow therapeutic drug, and on what basis is it classified? For instance, is an index strong inhibitor drug, is this classified if it causes more than a 5-fold change in AUC?
I think the question regarding that they may be confusing it with drug interaction, how we determine strong inhibitors. I would not, I would just define narrow therapeutic index drug for the purpose of assessing the BA study. And as I mentioned in my slide, generally we define narrow therapeutic drugs as those where we see small differences in dose or blood concentrations that can lead to serious either therapeutic failures or serious adverse drug reactions. And some of the examples I listed were warfarin, digoxin, etc. So for these NTI drugs, knowledge of AUC or Cmax, the exposure metrics, are critical for safe and effective use of drug product. So that's how the characteristics of an NTI drug are. And I think that's all I wanted to say. And the strong inhibitor drug, I think it's not relevant to BA assessment.
Thank you for responding to that question. We've got a couple more questions for Dr. Vaidyanathan, and here is the next question. What is the ideal washout period?
Ideally, the washout period should be 5 half-lives of the drug. Thank you.
Thank you for responding to that question. I've got another question for Dr. Vaidyanathan, and here is the question. Truncation is not allowed for high variable drug products. Is that high variability for absorption parameters or elimination parameters?
Thank you. Yes, truncation is not recommended for highly variable drugs. And in general, there are a number of factors that can contribute to high variability. Some of the factors for example, in the gastrointestinal tract can contribute to variability, which include, for example, gastric emptying, the intestinal transit time, the pH, or even presence or absence of food. So in addition, there may be other factors that can cause high variability, for example, extensive pre-systemic metabolism that can occur either in the lumen of the GI tract or in the intestinal cell mucosa, or even due to hepatic first pass. So, so there are many such factors that can influence the PK of the drug substance. Thank you.
Thank you for responding to that question. We do have one more question for Dr. Vaidyanathan, and here is the question. Please confirm or correct my understanding that the percentages defining high variability, i.e. greater than or equal than 30%, is the coefficient of variability.
I think you may be confused by the within subject variability versus the between subject variability. So generally, highly variable drugs are those that have greater than or equal to 30% in within subject variability. That is the variation in the same subject taking the same drug in 2 different occasions under similar conditions. So that's how we define highly variable drug. Thank you.
Thank you for responding to that group of questions. Moving on to our next panelist, we have a few questions that came in for Dr. Kofi Kumi, and here is the first question for Dr. Kumi. For a steady state study, is it required that we prove BE on Cmin along with AUC, tau, and Cmax?
Thank you for the question. I would say this would be a case-by-case, on a case-by-case basis. An example that I gave that they had to prove BA or BE on the same name, exposure response analysis has shown a correlation between reduction in seizure and same name. So that was that was recommended. So this, I would say this will be on a case-by-case basis, depending on if there's a special response or depending on other factors. So it's advisable to consult with the review division and have a common understanding when doing it. Thank you.
Thank you, Dr. Kumi, for responding to that question. You're still coming in a little bit fuzzy, so the tech team's going to reach out to you again, and we're going to move on to one of our next panelists, Dr. Akpol Aradiri. And here is the first question that we have for Dr. Aradiri. What in vitro tests can generate data that are considered surrogates of in vivo bioavailability in humans?
Well, the only one, the only in vitro test that can serve as a surrogate for bioavailability and bioequivalence is dissolution. Dissolution is the only in vitro test or quality test that can be correlated with in vivo systemic exposure. Thank you.
Thank you for responding to that question. Since you talked about dissolution, we have a question, and here's a question on in vitro dissolution for F2 studies. What pHs are recommended to perform the dissolution studies, and how many tablets will be used at each condition?
I believe in one of the slides, I think one of the slides has the pH values, and usually the pH for testing alcohol dose dumping usually is pH pH 1.2, which is equivalent to 0.1 normal HCl. That is the recommended pH in general. But if your dissolution methodology uses a dissolution medium that has a different pH, then you should in fact conduct the alcohol dose dumping studies first in 0.1 normal HCl using all of those alcohol concentrations, and then you also use your the dissolution medium that you are using in your regulatory method or your optimal method for that purpose. So you will do two sets of experiments, one with 0.1 normal HCl, which is at pH 1.2, and then your regulatory method in the dissolution medium in your regulatory method if it is not 0.1 normal HCl. And then in terms of sample size, it's 12. We expect 12 units to be used for each experimental run. Thank you.
Thank you, Dr. Aradiri, for responding to that question. We've got a couple more questions that just came in for you, and here is the next question. For a post-approval formulation change, if the BE is demonstrated in the pilot RBA study, is there a need to do a BE study? Could you also please share case studies and regulatory acceptance studies worldwide?
Thank you, Dr. Ford. I think the last part of the question is probably outside the scope of our program today, of our webinar today. I'm not quite sure that I can address that for the entire world at this time. But if you have demonstrated, if in fact in a relative bioavailability study that you've done, whether it's a pilot study or not, depending on, of course, what the batch size is and whether that's a final formulation. If that is a final formulation and you've done a pilot study, in a pilot study you've demonstrated adequate bioavailability, or perhaps even meeting the 90% confidence intervals on AUC and Cmax, or the AUCs and Cmax, I do not believe that there is any requirement or any need to conduct another BE study that you label as pivotal, unless there was a change that was made between the pilot formulation, pilot study product and the product that you're now expecting to go move forward with or to commercialize. But I do not believe there is any need to conduct a second study. Thank you.
Thank you for responding to that.
And perhaps, sorry, Dr. Ford, maybe I also want to quickly add that if in the pilot study you've demonstrated BE, as the question says, of course, that would indicate that, in fact, you have sufficient power, statistical power, to demonstrate, to see that you could calculate the confidence intervals, and that's meaningful. But if you do not have sufficient power for that purpose, then you can, even in that situation, you may want to give an explanation as to why you did not meet the confidence intervals. But conducting two studies on the same product with the same formulation is unlikely to be necessary. Thank you.
Thank you for responding to that question. We have just one more question that came in just a second ago for you, Dr. Aradiri, and here is the question. Could a biowaiver be granted for a lower strength of a BCS2 weak-based drug if the drug release is greater than 85% in 15 minutes and F2 similarity could not be performed?
This is not a straightforward question, but I will. My response is that if it is in fact a weak base, and and it's a BCS class 2, and you are having a dissolution test in which the product is releasing more than 85% in 15 minutes, I'm wondering whether or not that's actually an appropriate dissolution test for your product. If it is BCS class 2 with low solubility, one would expect that the dissolution of your product in a properly developed dissolution test would release up to 85% over a longer period of time. But in the event that, in fact, your product, if the dissolution method is assessed and found to be acceptable for whatever reason, if, for example, if you didn't use surfactants and all that, if the method is assessed to be acceptable, then yes, of course, even if you do not even if you cannot calculate F2, as long as all other conditions for the biowaiver request are met, the biowaiver may be granted. It's likely to be granted, even for a BCS2 product. But the key in this case is the additional information I would have liked to have is why you are having 85% dissolved within 15 minutes for a BCS class 2 drug. Thank you.
Thank you, Dr. Aradiri, for responding to that group of questions. We're going to check back in with Dr. Kofi Kumi. And here is the next question for Dr. Kumi. When should you evaluate PAUC and at what time points when assessing relative bioavailability for oral products?
Thank you for the question. I hope it's a little better. My audio is a little better now. Assessing partial AUC should depend on the clinical meaningfulness of the partial AUCs. Basically, the clinical response at what time, like giving in the sense of pain medications, you want quick pain, so usually Tmax is a good time for, it's a good time point for the first partial AUC, then sustainability. In ADHD drugs, as I said, you want a time point when there's a response early and then also sustain of the response. So it depends on the clinical usefulness of the partial UC of the time point to select and comparing the partial UC. Again, advisable to consult the review division to be able to come to a common understanding of what will be recommended. Thank you.
Thank you, Dr. Kumi, for responding to that question. We do have a little bit of static coming in, and we're asking you if you could speak up just a tad louder. I'm getting a response from SBI Central to try to check in with another presenter right now so that we can, the tech team can reach out to you. And we're going to swing back in with Dr. Chilukuri for another question that just came in. And here is the next question for Dr. Chilukuri. Are pilot studies always needed to be conducted prior to a relative BA/BE study?
Thank you. So pilot studies are not required per se, but are strongly recommended for the reasons that were delineated in the slides, primarily to get a better handle on the main relative BA study. So the answer to the question is no, they are not required per se, but strongly recommended. For example, the sample size question, for example, that was discussed earlier, that could be answered during the pilot study. So if the sample size question could be further evaluated in a pilot study, and based on the results of the pilot study, the main PA study could be better designed with an adequate number of subjects. That's one example. Thank you.
Thank you, Dr. Chilukuri, for responding to that question. We do have a few more questions that came in for you, and here is the next question, and it regarding AUC 0 to T. Do we need both AUC 0 to T and AUC 0 to infinity, I believe it's NF, infinity, as total exposure? And they're asking which one is primary?
Thank you for that question. So the guidance recommends both AUC 0 to T and AUC 0 to infinity as measures of total exposure. And the guidance actually does not specifically say which one is primary. So the confidence interval approach is to be applied to both those parameters for the determination of bioavailability. Thank you.
Thank you for responding to that question. We do have one more question for Dr. Chilukuri. And here is the next question. Can validated PK modeling approach be used to establish BA between a test IV formulation and a reference oral formulation?
Thank you for that question. So I cannot say that this is very common. The most commonly seen approach is not the population PK modeling approach. So I think this should be taken to the specific division for further discussion, because a majority of the cases what we see are straightforward non-compartmental analysis comparison between the two formulations. So cannot say that this is very common. This could potentially be discussed with the review division for further advice. Thank you.
Thank you for responding to that question. Moving on to our next panelist, we just got a few questions that came in for Dr. Jayabharathi Vaidyanathan. And here is the next question for Dr. Vaidyanathan. my listed drug A is an IR tablet. The product under development B is also an IR tablet for a different indication, and it's given a 505(b)(2) application. Should we perform a single-dose bioavailability study to understand the PK parameters, or can I simply rely on in vitro dissolution data?
Okay, so as I understand, you are developing a IR tablet for a different indication than the approved listed drug, which is also an IR tablet. So in this case, generally, we would recommend that single dose fasted bioavailability study be conducted to compare the two products. And generally, in vivo study would be needed, and I'm not sure you can use in vitro dissolution data in this case. Thank you.
Thank you for responding to that question. We've got a couple more questions that came in for Dr. Vaidyanathan, and here is the next question. What does the truncated AUC mean?
As you all know, for bioavailability studies, usually we want the area under the curve and the maximum plasma concentration, the Cmax, measured. An estimation of AUC requires frequent blood sampling over the profile of the drug, and usually this includes both the absorption and elimination profile. For long half-life drugs, sampling for long periods of time to get the complete PK characterization may be quite difficult. And to overcome that, we have used truncated AUC provided that the drug product at a suitable time point, provided that the drug product has completely moved through the gastrointestinal tract so that complete absorption of the drug substance can be obtained. So that's a suitable time point should be selected for truncated AUC. And as these are complex situation, probably contacting the review divisions would be appropriate.
Thank you for responding to that question. We're going to check back in with Dr. Kofi Kumi after the mic adjustment and see if we are able to get a question out to Dr. Kumi. And here is the question. What type of pharmacodynamic marker can be considered as endpoints for a BA assessment?
Thank you for the question. The TB marker must be related to the disease process of the disease condition. It should be justified. That is it's appropriate. Biomarkers usually should be validated or qualified and should be appropriate. It should be used. Thank you.
Thank you, Dr. Kumi, for responding to that question. We have just under a minute left. We're going to try to squeeze in one more question if you're able to answer this one. And here is the next question. What kind of clinical trials can be used in evaluating BA for topical products?
Thank you again. Basically, the clinical trial can be a phase 2 standalone trial, or it can be a subset of a phase 3 trial where you select endpoints and evaluate it during a subset of subjects during the phase 3 trial. So it can be a standalone in phase 2 or a subset of phase 3 trial. Thank you.
Well, that's all the time we have for questions. A huge thank you to our panelists for your very informative and timely talk, and also for responding to numerous questions that came in. We do just have a few short SBIA reminders. This activity is eligible for an attendance certificate, which supports continuing education by SOCA, RAPS, SQA, and ACRP. Please refer to our website at fda.gov/CDER SBIA for more details.